have a complete strand. So, as I mentioned before, you're going to have leading and lagging strands. So there's your helicase, single strand of bonding proteins, growing replication fork. 3 prime, 5 prime right beneath it, 5 prime on the end, and then 3 prime on that bottom end. So we have our DNA polymerase 3, which is again the enzyme that's going to add the nucleotides, going in this direction, 3 prime to 5 prime. Okay, we're going in the right direction, 5 prime to 3 prime. So we can just make a nice big long strand. But if we go down here, 3 prime to 5 prime, it doesn't work. This is what we just proved on the last slide. So we have to go in the other direction, 5 prime to 3 prime here, and then a shorter segment, and a shorter segment. So there's the limits of the DNA polymerase 3. It can only build onto 3 prime end of an existing DNA strand. So we call these Okazaki fragments. So after Okazaki over here, Japanese scientist. So then we use ligase to connect it, and again we call this one down here our leading strand. And the leading strand is a continuous synthesis because it's going in the 5 prime to 3 prime direction. The lagging strand, you'll see, is sequenced by segments of Okazaki fragments joined by the ligase. So we call this the spot welder enzyme. The ligase is the spot welder enzyme. So let's go ahead and look at the replication fork and the replication bubble. 5 prime to 3 prime for the top strand, and then the bottom 5 prime to 3 prime as well. So as we start to open it here, we're going to have direction of replication that are opposite of each other. So one side will be the leading, the other side will be the lagging. Let's go ahead and look at this. DNA polymerase 3 will go in that direction, 5 prime to 3 prime. Okay, that will be the leading strand. Down beneath that, you're going to go 5 prime to 3 prime in the other direction, and that will be Okazaki fragments that create the lagging strand. Okay, as we make it a little bit larger so we can see it, growing replication fork in both directions. Again, here's our leading strand, followed by the lagging strand of Okazaki fragments. Then on the other side, we are going to have a leading strand, okay? And don't be confused, that's because we actually started right here. It's not that this switches. If you know, this is kind of the after, and here's the before, right where we split. So that's why this side, so it's from where the replication fork starts. As long as in this direction, it will be 5 prime to 3 prime. But from here, this would be 3 prime to 5 prime, which is why we have to come in this direction. Okay, so the other side is the same way, 5 prime to 3 prime of lagging strands. So in any given replication bubble, you're going to have 2 leading strands and 2 lagging strands. So the start in the DNA synthesis, we have to have these things called RNA primers. They're what actually start the whole sequence off. So again, the limits of the DNA polymerase 3 can only build onto the 3 prime end of an existing DNA strand. So let's go through and look again. Here we add a primase, little segment of RNA. And then the DNA polymerase 3 can sort of hit the ground running. So you can kind of think of the RNA here as kind of like a runway. It's sort of letting the plane catch up speed before it can finally take off, as maybe a poor analogy. So on the other side, you're going to have shorter segments of RNA as well, RNA primer. Okay. Then RNA primer is built by an enzyme called primase. It serves as the starter sequence for DNA polymerase 3. So now we have to replace the RNA primers with DNA. So here you have the ligase, which is going to, and the DNA polymerase 1 together. The ligase sort of connected them, and now we have the DNA polymerase 1, which is going to take away the RNA primer. So you can see it going away. There goes the RNA primer. Here's the other one and the third one. okay? And the fourth one down here on the leading strand. So DNA polymerase 1 removes sections of RNA primer and replaces with DNA nucleotides, okay? So, but DNA polymerase 1 still can only build onto 3' ends of existing DNA strands. So, chromosome erosion, we have a problem here. Okay, all DNA polymerases can only add to the 3' end of an existing DNA strand. So again, you have your DNA polymerase 1, but down here on the leading strand, we have a segment that we can't reproduce because the DNA polymerase 3 can't add to such a small segment. So what happens? Well, from the 3', there's a loss of bases at 5' ends in every replication. Chromosomes get shorter with each replication, and this limits the number of cell divisions. All right, well, it seems like we're losing DNA, essentially, every time we replicate our DNA or our cells. So, kind of a problem. So how has evolution sort of adapted to this problem? Well, let's look. We have something here called telomeres. So telomeres are repeating non-coding sequences at the end of the chromosomes. Essentially, they are a protective cap. So non-coding means that the DNA sequence is not converted into RNA, which is not converted then into proteins. So you're not going to make anything functional out of a telomere. You're not going to make protein out of it. So the point of this is it's essentially limiting cell division to around 50. And let's see what happens here. So we have this huge sequence of repeating DNA. And telomerase comes through, which is another enzyme that extends the telomeres. And it can add DNA bases at the 5' end. And it's a different level of activity in different cells. So this is where you're going to see that there's telomerase is high in stem cells and in cancers. So why would this be? Why would you have a high level of telomerase and in stem cells and cancers? Well, what we're finding is that this telomerase is essentially kind of keeping the replication of cells going on a consistent rate, which is essentially what cancer is. It's sort of a rogue cell line that's continuing to divide even though it shouldn't. So we'll have a little bit more on how cancer is related to telomerase in another PowerPoint. Let's continue to move on. So let's have a quick review here. Again, the direction of the replication is, in this case, is left to right. Here's our helicase, single strand of binding proteins. Here's primase, which is going to add the RNA primer. DNA polymerase 3, which is on both sides. Okazaki fragments, which are on the lagging strand. Ligase, which is connecting the Okazaki fragments. DNA polymerase 1, which is going to go through and take out the RNA primers on the leading strand down here and the lagging strand up above. So the DNA polymerases, let's go ahead and look at these. DNA polymerase, it's going to change out 1,000 bases per second. This is the main DNA builder, DNA polymerase 3. So DNA polymerase 1, it does 20 bases per second, so much slower rate. And its point is editing, repair, and primer removal. So DNA polymerase 1 was discovered by Arthur Kornberg, who received the Nobel Prize in 1959. He has actually two sons, Roger Kornberg and Thomas Kornberg. Roger Kornberg received the Nobel Prize in 2006. Thomas Kornberg also did a lot of work with sort of characterizing and further defining the role of both polymerase 1 and polymerase 3. But Roger Kornberg received the Nobel Prize in 2006 for his work actually with transcription. So let's move on. Editing and proofreading DNA. So if you look at the top, we have a thymine dimer, distorts the DNA molecule. So a thymine dimer is when you have two thymines that are bound to each other, and you can see how that might be a problem. And so a nuclease enzyme cuts the damaged DNA strand at 2 points. Number 3, repair synthesis by a DNA polymerase fills the gap. And then, number 4, DNA ligase seals the remaining nick. So we have fixed that thymine dimer. So there's 1,000 base pairs a second, and you're going to end up with lots of typos, such as the thymine dimer. So the DNA polymerase 1, it proofreads and corrects the typos, and it repairs the mismatched bases. And this removes the abnormal bases, and it repairs damage throughout life. So this reduces the error rate from 1 in 10,000 to about 1 in 100 million bases. So it's fast and accurate. It takes E. coli, which is a type of bacteria, less than an hour to copy 5 million base pairs in its single chromosome. And it divides to form 2 identical daughter cells. The human cell copies its 6 billion bases and divides into daughter cells in only a few hours. So it's remarkably accurate, and there's only about 1 error per 100 million bases, or about 30 errors per cell cycle. So what does it really look like? Well, again, this is happening at not just one point, but multiple points. You can see they have the sort of the silhouette of the DNA on the right, and there's actually 4 different replication bubbles. So hopefully this is clear. If not, come to class with any questions.